I'm Tony Stretton, and I'm a professor in the zoology department, and I'm also got a major commitment to the neuroscience training program, because now what I do is neurobiology. In the past, I've been a chemist and then a molecular biologist when molecular biology meant working on the genetic code, which is what I did with Sidney Brenner and Francis Crick way back. With colleagues, I worked on the collinearity of the gene and the polypeptide chain. So I was a protein chemist, so I did the protein bit, and Sidney Brenner actually did the genetics bit. So that was showing that the structure of the gene and the order of mutants in a gene corresponded to the effects on the polypeptide chain. So that was this collinearity problem, which was a big deal way back, but it's so obvious now no one's interested anymore. And then the other thing we did was to work on the chain terminating codons. So we decoded the amber and ochre and opal codons. And then the genetic code was solved, the rest of the genetic code was solved by other people. So at that time it was like the Holy Grail was trying to figure out what the genetic code was, and then so when it was done, people thought, oh, what are we going to do now? It's like the world's come to an end, and there's nothing interesting left, which of course is utter nonsense. But anyway, that led to people moving out of molecular biology, as it was then, into other fields, and I was one of them who moved to neurobiology. So I've been working on how the nervous system of a very simple animal, nematode worm, work on a big one, so not Caenorhabditis elegans, which all of you in genetics know well, right? But Ascaris, which Ascaris suum is called, it comes from a pig. It's about 20 centimeters or 30 centimeters long. So it's got big cells, so you can do electrophysiology. And you can also do single cell biochemistry. So we're interested now in finding out how it works, and so being a simple system, I thought we'd knock it off in 5 years, because it's only got 298 nerve cells. But that was 50 years ago, and we still don't understand it. Is to do a variety of things, so you can find out what suits your particular personality, both your mind and your manipulative skill. I love using the microscope, and so anatomy, structure, that's what anatomical structure is at the basis of almost everything we do. So it has to be related to the actual structure. So I really like sitting in the microscope and figuring it out. And then I also enjoy gazing at sequences. The other thing is to find somebody whose mind you admire. So find someone whose mind meshes with yours and excites you. Graduates, you work your ass off, and you've got to work hard. You've got to be committed, and to be successful in research, it's a very demanding game. I think I'm lucky if I have a buzz of excitement twice a year, and the buzz lasts about 5 minutes, so you've got to have some other reward system in there, and what it is for most people, I think, is pleasure in the actual doing of it, so you have to enjoy the process. So as an undergraduate, take the opportunity, which is huge in this university, to find, to sample different sorts of research.